discussion about biotechnology and genetic engineering. I think I would have given this as the principle of biotechnology, which is nothing but DNA 1 plus DNA 2 will give rDNA. I hope you remember that our target was to get the insulin hormone, so we consider that as an example through which we will be discussing the various biotechnology concepts. So for which there is a need for insulin gene, Ig, which stands for insulin gene, plus there is going to be a plasmid, which is going to be a vector, will give recombinant insulin plasmid. Okay, so I request people who haven't gone through that particular introductory video, so please do have a visit, okay, and then come back to this video so that you will have continuity. Okay, so and we are going to get this plasmid from a biotechnology company. So I have said why are we going to do that? So now the question is how am I going to get this insulin gene, right? So this was the question which was posted in the previous discussion. So the answer is, you have various thoughts might come to your mind. So how are we going to get this insulin gene? So some might think that we can go to the pancreatic cells, that's beta cells, where naturally the insulin hormones are produced. So from that we can get the insulin gene. So various options, various possibilities might come to your mind. So before this, again, to understand these things with a clarity, you are supposed to have a clear-cut distinction between gene, DNA, etc., for which I have given a separate video. So in case you have any confusions, please do visit that particular video and then come back to this discussion. Right. Now, one concept that I'd like to say is that DNA, the double-stranded helical structure, which is or the chromosomes condensed form, which is there inside the nucleus, is going to be same for a person. So if you consider me, I have n number of nucleated cells, so except RBCs and platelets, matured RBCs and platelets. So all my cells are nucleated, which means that inside the nucleus there is going to be a DNA in the form of chromosome. So if I am going to get the DNA from, say, a thyroid cell, from a pancreatic cell, a particular beta cell, the DNA profile in these two cells are going to be similar. So there's no difference in the DNA profile. So this has to be kept in mind. Now, in order to get this insulin gene, so I have to know where is this insulin gene present. The insulin gene is present in the DNA. So if I'm going to consider this as the structure of DNA, right? So this is in turn made up of n number of genes, okay, and each gene is going to code for a particular protein, of which I might consider this to be an insulin gene. So this might be the insulin gene, right? Now, for me to get this insulin gene, I have to get this DNA, which is there inside the nucleus in the form of chromosomes, right? Let this be the chromosome in the form of chromosomes. So the best strategy what we have here is, in order to get the insulin gene, pick up a cell. What cell? It has to be a nucleated cell. Why a nucleated cell? Because nucleated cell is the one which is going to have the DNA in the form of chromosome. Right. So which cell? From where? What hormone do we need? We need insulin hormone from human. So we have to pick a particular nucleated cell from human. get the DNA out, right? And from there, we have to specifically target your gene of interest. So this is the strategy. So by which you will be clear that you did not specifically get into the pancreatic cells. That is it, the DNA profile is going to be same in all the cells. So the best cell, the easy cell, the nucleated cell that we can get for this purpose is blood cells, right? So blood cells has got RBC, WBC, platelet. There are going to be three cells: RBC, WBC, platelet. Of which you all know that RBC, matured RBC, which is there in circulation, has got no nucleus. Platelets has got no nucleus. So whereas you can very well concentrate on white blood corpuscles. So you can easily isolate some 2 to 3 ml of blood, get the WBC out. There are procedures for it. Now which will not be, which are on the point to be discussed here. So get the WBC out. WBC has got good concentration of DNA. Get the DNA out, okay, in the form of chromosome. In that you will be able to find insulin gene. So the one thought that we are supposed to, one concept that we are supposed to have with clarity is, right, the DNA profile in each and every cell is going to be same, right? Whereas some gene gets expressed in some cells. For example, insulin gene is there in WBC, but it is not getting expressed. The same insulin gene is there in the beta cells of pancreas, where it is getting expressed. So we tend to see the insulin hormone in that cell. These are all some further basic concepts. If you have any doubt in these things, you can kindly let me know. I'll be able to give a video on that. Okay. So now, again, to summarize, I need this insulin gene, which I cannot get it alone. So I know that the insulin gene is sitting inside my DNA. So my plan is get the DNA out of the cell, right, and concentrate on insulin gene and put it in plasmid, where I get recombinant insulin plasmid, through which I am going to continue further my process with insulin hormone production, right. So this particular discussion is going to talk about DNA isolation from cell. DNA isolation. So this DNA can be isolated from any cell provided the cell has to be a nucleated one, the nucleus. So that's the criteria. So here, since we need a human insulin gene, our target cell here is a human cell, of which the convenient cell that we can access conveniently is a blood cell, in which we are going to concentrate on WBC, since that other cells doesn't have nucleus, right? So I'll have a nucleus, I'll have a cell here, right? Now, if we look into the basic structure of cell, that's going to be a nucleus, a cell, nucleus, and you know that the space between the nucleus and cell, that is outer membrane. So this is going to be the membrane, outer membrane. Is going to be the cytoplasm, right? So here is what our DNA is sitting inside. So we have to get this DNA out. So for this, the strategy is first break open the outer membrane, right? So once the outer membrane is broken down, automatically the nucleus will also disintegrate, so that the DNA gets out of the nucleus. So what we can do, we have two strategies. The first strategy is, two steps. The first step is break open the outer membrane. So how are you going to do that? So first we have to know what this outer membrane is. So outer membrane is going to be constant. It is going to be the plasma membrane, whatever be the cell is going to be, whether it's going to be plant cell or whether it's going to be an animal cell or it's going to be the fungal cell or bacterial cell, right? So plasma membrane is going to be constant because cell membrane is there in all cell, but except animal cell, plant cell or a bacterial cell or a fungal cell, it's going to have an additional membrane which is called a cell wall. Okay, so you have to design your, there's something called as cell lysis solution, or sometimes since it's going to maintain pH, we will also call it as a buffer, cell lysis solution or buffer. We are going to make this, and the constituent of this cell lysis solution depends on the cells from which our DNA isolation is being planned. Okay, so in case if you are going to plant an animal cell like human cells, what we have taken for our present discussion, you can just add some detergent. Right? Detergent is because our cell membrane is made up of this phospholipid bilayer, so detergent will easily break down the plasma membrane. So plasma membrane will disintegrate. Whereas if it is going to be a bacterial cell, what is bacterial cell made up of? There is going to be a cell wall surrounding the plasma membrane, for which you have to target the cell wall. So what is the cell wall, bacterial cell wall made up of? It is made up of peptidoglycan. So you have to add lysozyme, because lysozymes are the enzymes which are known to disrupt the the cell wall synthesis. This peptidoglycan, N-acetylglucosamine, N-acetylmuraric acid. Right, so if in case the point is, in case if this is going to be a bacterial cell, you have to add lysozyme. In case it is going to be a fungal cell, again fungal cell has got a cell wall, so you have to add your cell lysis solution should have chitinase, because fungal cell wall is made up of chitin. In case this is going to be a plant cell, what is plant cell? Plant cell wall is made up of cellulose, so your cell lysis solution should have cellulase, right? So your cell lysis solution or cell lysis buffer constituent or composition will vary based on the type of cells that you are planning for. So in case there is going to be an animal cell, I repeat, since there is no cell wall, only plasma membrane is there, you can just simply add a detergent which will disrupt the cell plasma membrane, right? So I hope this is the first step, right? So you have a tube, you have put your cell and add cell lysis solution, right? Cell lysis solution. So I have given enough discussion about what cell lysis solution is. Sometimes we even call it as a buffer, because the role of this solution is to maintain the pH. I hope your chemistry would have taught you what literally a buffer is. A buffer is something which will try to maintain the pH in a particular range, within a particular range. So once you try to target this membrane, once the DNA comes out, the point is your DNA. Our idea is not only to get the DNA, but also to get the DNA in a very intact form. Right, as it is, because you would come to know that even a single nucleotide in the gene, insulin gene, which is there inside the DNA, should not get disturbed, right? So for which the pH maintenance is very much required. So we are going to provide a buffering capacity for this solution. So this is the thing. So once you add this, your DNA comes out. So your solution, your lyse cell, will not only give the DNA, but also the organelles, protein, everything sitting, because cell generally has lots and lots of component. So every component will get out of the cell, but our idea is to target only the DNA. So the second step is we are going to add. So this is going to be the first step, cell lysis. Second step is we are going to add DNA concentrating, okay, agent, or you can also call it as DNA precipitating agent. So which is generally chilled alcohol, preferably it is going to be ethanol. So this question was asked many times in NEET exam, especially this 2021. The DNA precipitating agent which is used in DNA isolation is going to be chilled ethanol. So once you add chilled ethanol, your all the component will remain as it is, whereas your DNA will flow. So you can get this DNA easily into another tube, right? Now you have got your DNA ready with you. Now, I hope this is clear. So there are just two steps: break open the membrane, release the DNA from the cell, and isolate the DNA by adding a DNA precipitating agent or DNA concentrating agent, which is nothing but the chilled ethanol. So that's it. So the next thing is, how are we going to isolate or point out this insulin gene from whole the list of genes there in the DNA? Because I said that DNA is going to have n number of genes, of which our gene of interest is going to be insulin. So the next video discussion will be especially oriented, okay, towards understanding the technique that is involved in DNA gene isolation from the DNA, right? So keep watching. Please do subscribe. In case you have any query, let me know. Thank you so much.